Hi, everybody. Today we are going to learn about aerobic cell respiration. We have already learned about anaerobic, we learned about glycolysis, and we learned about fermentation. And today we are going to learn about what happens when oxygen is present and if organisms can use that oxygen. So we're going to start today by talking about the first and second parts of aerobic respiration, which is the process of glycolysis and the Krebs cycle. So first of all, let's review. We already know that the first step of any type of cell respiration is glycolysis. And we already know that glycolysis happens in the cytoplasm. And we also know already that glycolysis is going to break glucose in half into two pyruvic acids. And as a result of this process, we get two ATP molecules. Those ATP molecules are good for energy. We get two molecules of NADH. They are holding energetic electrons. And we get two molecules of pyruvate. And those pyruvate have more potential energy that can be broken down. Here is the process of glycolysis: pyruvate, ATP, NADH. All right, now what is next? What is after glycolysis? Well, we have something called the Krebs cycle, which is what we're going to be covering today. And after that is the electron transport chain. So if oxygen is present, that means aerobic. The pyruvate is going to move to the mitochondria. This is a picture of mitochondria. Where was the pyruvate before? Yeah, as we know from previously, the pyruvate before was in the cytoplasm, and now it is in the mitochondria. So the next step is the Krebs cycle, which is also sometimes going to be called the citric acid cycle. This is happening in the matrix of the mitochondria. What is that, the matrix? The matrix of the mitochondria is just the inner part on the inside. Here it looks blue. The matrix just means space. It's the inner space of the mitochondria. What is the Krebs cycle going to use? We're going to start with pyruvate, and we're going to make a bunch of things here. Let's define what these things are before we continue, because you do need to know what these things are for. First of all, we're going to make NADH. We already know that. That is carrying energetic electrons. And then we're going to make FADH2. FADH2 we haven't talked about before. It is almost the same thing as NADH, right? It's almost identical, and it is also an electron carrier. We're going to make two measly ATPs. That's not a lot, but it's something. And then we are going to make a waste product, CO2. CO2 is carbon dioxide. What do animals do with carbon dioxide? Where does it go? Right? We exhale carbon dioxide from our lungs into the air because we don't need it. So CO2 is a waste product. Okay, so I want to skip to this picture because this picture is going to show us what is happening with the Krebs cycle. We don't need to know the details of the Krebs cycle, but I do want to show you what it looks like. We are going to start off with pyruvate, and pyruvate has 3 carbons. And right away, pyruvate gets turned into acetyl-CoA. It loses a carbon. And then acetyl-CoA is going to undergo all of these crazy changes. If you see, acetyl-CoA is going to be added to something else, and it's going to create this molecule called citrate. And then it's going to undergo another reaction, and it's going to become a slightly different molecule called isocitrate. And it's going to change like 10 more times. The pyruvate is going to keep changing forms. It's a chain of chemical reactions. Now, why does that matter? Why are we changing pyruvate? Because the cell can take these chemical reactions and use those chemical reactions to make other things as well. One thing that you can see here is that the cell is making NADH, right? What is NADH holding? It's holding energetic electrons. So this chemical reaction here is making NADH. NADH is stealing these electrons from this isocitrate molecule. It's taking the electrons away. In the process, we're making a waste product. And then more chemical reactions happen, and more NADH is made, and more carbon dioxide waste is made. If we look down here at the bottom, we're making some ATP as a result, right? This is where ATP comes from. This is the FADH2. Remember, it's basically the same thing as NADH. And then we're making even more NADH. So pyruvate is getting broken down little by little, step by step. It's getting broken down, it's getting changed. And we are producing CO2, we're producing ATP, and we have electrons being held in NADH and FADH2. So that is the Krebs cycle, a very short explanation of the Krebs cycle. So what is the function? Why do living things undergo the Krebs cycle? The reason is right here that I am highlighting. It's to make our electron carriers. Those are very important. All right, what else are we making? We're making a little bit of ATP, but it's not very much. It's 2. It'll maybe, keep you going for a very short time, but it's not enough. It's not enough for living things. And then we're also producing this waste product. We don't really care about CO2. It is just a waste product. All right, so let's figure out if we learned anything from today's lecture. So our first question, what is the purpose of NADH and FADH2? How much ATP is made by the Krebs cycle? And where does the carbon dioxide we exhale come from? So a few questions there to answer. Okay, so number 1, what is the purpose of NADH and FADH2? NADH and FADH2 carry energetic electrons. How much ATP is made? It's only 2, not very much, just 2 ATP. And finally, where does the carbon dioxide we exhale come from? It comes from the Krebs cycle. It comes from what used to be glucose turned into pyruvate, and then that pyruvate releases carbon dioxides. So where does the carbon dioxide we exhale come from? It comes from what we eat. The sugar we eat becomes carbon dioxide. Okay, we're going to stop there for now. Next up is going to be the electron transport chain. Let me know if you have any questions.